Hi everyone, welcome back to end of year practice session. So today we will start with the term one. The subtopic that we are going to discuss is enzymes. So how is going to happen this whole thing that I will be focusing on the KPIs and then the practice questions which are followed, and all the notes will be made on this whatever is related to the questions. So first KPI, it asks from you, recognize enzymes as biological catalyst. So we need to know that the enzymes are biological catalysts. So who is the catalyst? Catalyst is anything that increases the rate of chemical reaction, and make sure you also know without being used in the reaction. So why we say biological catalyst? Because all these are within the living body without being used in the reaction. Now enzymes are biological catalysts, and also one more thing that you have to know, that enzymes are protein in nature. Enzymes are protein in nature. Okay, so basically this is what we have to know. Let's go to the questions and we will see more notes. What comes up, I will be adding up. Use the diagram below which shows the catalytic cycle of the enzyme to answer questions 1 and 2. Okay, so question 1, complete the table below by writing the name of each labeled part in the diagram. So part A, so since this is an enzyme and this hollow space or a hole-like structure on the enzyme, it is called as active site. So structure A is active site. B is binding to the active site of the enzyme, so B is a substrate. Now remember that every single enzyme is very specific to substrates. So not all the substrates can bind to the enzymes. Why? Because they have complementarity between the A and B, like between the active site and the substrate which is binding to it. So how we complete this equation? This complete equation is called E plus enzyme plus S. So E plus substrate, enzyme plus substrate. It leads to E dot S complex. Here it's lead to processing. So here the processing or catalytic reaction is happening, and then it lead to E plus P. Now here C is the P, and what is the P? P is a product. So we are going to write over here C is product or products, whatever you say. Okay, let's move to the next question. Explain what is happening in each step as shown in the diagram. I have explained you already, like what's happening over there. You can say the same in your own words. Okay, so in step number 1, in the first stage, what you can see, the enzyme binds to the substrate. Okay, so it's E plus S, then it leads to the formation of ES complex, and then it leads to the processing or catalytic reaction or chemical reaction, and then in the end you say that it leads to the formation of the product. So you have to say it in words, but I can write over here E plus S gives you E dot S, gives you E dot S complex, and it gives you E plus P. Okay, so you know what is S and P, and this is known as the enzymatic cycle. Question number 3, which of the following explains why enzymes are specific for certain reactions? So how you say that the enzymes are very specific for certain reactions? We already said that enzymes are the one that bind to a specific substrate, so you should know about it. And why they do this? They have active sites. Okay, let's read the options. The active site of the enzyme, the active sites of the enzyme binds only with certain substrates. Yes. The enzymes are consumed only with certain reactants. No. Enzymes are never consumed. They just bind and then being produced at the end of the reaction. Enzymes are made up of special type of carbohydrates. No. I told you they are made up of proteins. Enzymes are made in the cell with specific processes. It's not necessarily that the enzymes are made within the cell. You have them from outside as well. and so this statement is a bit vague, so we are going to cross it out, and A is the answer. Next KPI, please. Explain using graphs. So now in this part, you expect that you might get a graph and how enzymes lower the activation energy. So this is another point now we know. The point number one was enzymes are protein in nature. Point number two is enzymes increase the rate of chemical reaction. So 1, 2, okay. And then here we learned point number 3, that enzymes are very specific to the substrate. Now we are learning the property number 4, that enzymes are the one who lower the activation energy of the reactions. And what they do after, you can see a catalyzed and uncatalyzed reaction. Okay. In the graph, which of the following numbers represent the activation energy of a reaction before? So here it is, be very careful, it is before the addition of enzyme. So this is the graph that shows you progression of the reaction is going this side. You have the products, you have the reactants, and the reaction goes 1, 2, 3, and 4. Okay, so we need to see like what are these. So basically what he wants from you, it's like activation energy. So these two are the activation energies, number 1 and number 2. So the higher the activation energy is without enzyme, so that's what it says over here, enzymes lower the activation energy. Okay, so the activation energy which is less, so number 2 is with enzyme, this is the activation energy with enzyme, and number 1 is without enzyme. Okay, so how you say with enzyme? With enzyme mean it is a catalyzed reaction, and without enzyme it is an uncatalyzed reaction. Okay, so this is how you learn it. So before the addition of enzyme, it's number 1, but if they ask you after, so after the addition of enzyme, this is number 2. Okay, let's go to the next question. Demonstrate using visuals. So here we have some visuals, how an enzyme mediated chemical reaction occurs. You should know what is an induced fit model and what is a lock and key model, enzyme substrate complex, role of the active site, and enzyme specificity. We have already discussed what is enzyme substrate complex. We know what is an active site. We have discussed what is specificity, like every enzyme bind to a specific substrate. And what does the lock and key model means? So this diagram which is shown down is an example of the lock and key model. So here the lock is enzyme. Okay, so always remember X, for example, X is an enzyme. An enzyme acts as a lock, and this sugar on the top is a substrate, and substrate always acts as a key, and this key has a specific shape, and the lock has a specific hole in which the key can enter. So this is how the lock and key works. Okay, so this is enzyme substrate complex already done, and you can break it down. You can release the sugar into simple sugar molecules, you can break them out, and then you release the enzyme. So enzyme in the same amount it can be recovered. What is meant by an induced fit model of an enzyme? The enzyme structure is altered so that it can be induced to fit many different types of substrates. So when it says many different types of substrates, it's wrong. Why? Because it cannot fit many different type of substrates. It can fit only specific substrates. So it is against this enzyme specificity. That's why this is wrong. The presence of the substrate in the solution induces the enzyme to slightly change its structure. No, it doesn't have, substrate doesn't have to induce it to change its shape. This is wrong. The substrate can be altered so that it is induced to fit. No, the substrate cannot be altered. So this is wrong again. We are left with only D option, but let's read it also. The enzyme changes its shape slightly as the substrate binds to it. Yes, that's true. So substrate will stay same, but the enzyme can little bit change its shape to fit it. You adjust it. So that's the answer. Molecule X in the diagram. Okay, I already explained you. This is an enzyme. So answer is C. Which of the following is correct about the diagram? This diagram shows you an enzyme lock and key model, but we will see what's correct about the diagram. Molecule X can be used in any other reaction. It can be used in an other reaction, not any mean, okay, now you put it in with with lipids, or you put it with protein. No, we cannot do that. So this option is wrong. We go to the second one. It's not any, it's an. Molecule X is made up of carbohydrate. No, we already said it's protein. Then the reaction produces more energy in the presence of molecule X. No, we don't have to work on the production of energy or release of energy. In the presence of the molecule X, we only focus that molecule X, it lowers the activation energy. This is what we have learned, activation energy. Okay. The last option, that's going to be the answer, but since all three are wrong, the upper ones, the reaction will be slower in the absence of molecule X, because molecule X speed up the reaction. Yes, that's correct. Moving to the next page. The active site of an enzyme is the region. Okay, what is this region? You should know it's a region where a substrate binds. Okay, so or there are two molecules. One are called as regulators, and the second one are called as inhibitors. And inhibitors are further two types. Some can be competitive and some can be non-competitive. So competitive inhibitors are those who bind to the active site. They compete with the substrate to bind to active site. Okay, they bind to the active site, they compete with the substrate, while the non-competitive inhibitors, they don't compete with the substrate and they bind to another place or another, sorry, this is not side, this is site. 1 minute. Active site. And the non-competitive binds to another site than the active site. So the enzyme may have another site where the non-competitive inhibitors can bind. Anyways, coming back to the question, the active site of an enzyme is the region that binds non-competitive inhibitors to enzymes. That binds it binds not non-competitive inhibitor, it binds substrates. Okay, it is for the substrates, so this is wrong. It binds allosteric regulators of the enzymes. No, allosteric regulators are basically the one which are bound to the any other site of other than the active site. It is involved in the catalytic reactions of the enzyme. Yes, that's the correct answer. It is inhibited by the presence of a coenzyme or a cofactor. No, it's not inhibited by enzyme. It's inhibited by inhibitors, as I explained you in the side. So it is inhibited by inhibitors. Which of the following explains why enzymes are specific for certain reactions? So by the way, I'm just looking at down now. So the competitive inhibitors and non-competitive inhibitors are down. Okay, we will see them. Which of the following explains why enzymes are specific for certain reactions? Why enzymes are specific? We just said over here, because of the active site. The active site of the enzyme binds with only certain substrates. Yes, that's possibly the answer, but we move on. Enzymes are consumed only, not consumed, they just bind. Enzymes are made up of special type. Again and again it's coming, so remember it's protein. Enzymes are made in the cell with specific processes, as we discussed already. This is not the correct answer, so answer is A. Explain the effect of the environment, activators and inhibitors on the structure and function of the enzyme. Temperature, pH, activators, competitive inhibitors and non-competitive inhibitors, with an explanation of the allosteric site. Allosteric site is any other site than the active site. Okay. Which of the following is not a factor that affect the activity of the enzyme? Temperature, we just discussed up. If you look into here, we just said temperature is there. pH, yes, it is there. Amount of the enzyme, it can be actually one of the factor, and type of the product is not a factor. And what we are going to look for? Not a factor. It is a factor, it is a factor, it is a factor, it's not a factor, and we want to find the one which is not. So this is the answer. Type of the product is not affecting the activity of the enzymes. An enzyme that is found in the human gut was placed in solution of 90 degree and then added to a reaction to see its effect. The rate of the reaction did not change by adding this enzyme. Which of the following could be a possible reason for it? What's the possible reason? You know that all the enzymes work at optimum temperature. Optimum temperature. And what is the optimum temperature? Optimum temperature is the temperature at which every enzyme works at its best. Okay, optimum temperature is the temperature at which an enzyme works at its best. Okay, so you must remember, wait, let me see. Yeah, there is down. Okay, so we will see down. So for example, over here we have a graph, but this graph is related to time. Okay, so I will draw a graph, for example, like this one. You must remember the graphs like this. You have studied many times that here it is temperature, or let's say pH, and here it is the rate of the reaction. So how you find the optimum value? Yes, I can read your minds. You are just saying that you look for the peak and drop it down, and whatever value comes over here, it is the optimum temperature or optimum pH where the enzyme works at its best. Okay, now imagine it was like as a human. So if it is human digestive enzyme, okay, so what is should be the optimum temperature? Like the same as the body temperature, so it's around 37, 38, whatever. Now if you heat more than it, as it is mentioned over here, you heat it at 90 degree, what's gonna happen? Since we know that enzymes are proteins, okay, enzymes are proteins, and what will happen to these proteins? These proteins will denature, and when these proteins will denature, what happens? The active site shape will change. Active site shape will change. And now what does the active site do? It binds with the substrate, right? So no substrate can bind to it. No substrate can bind to it. When no substrate can bind to it, what will happen? No reaction, no product will be produced, and that's the whole logic behind the question. Okay, so the enzyme produced the reactants faster? No, of course no. Like we are saying that there is the product is not producing, so this is wrong. The enzyme changes the type of the chemical reaction? No. The enzyme became denatured, and this is what we just discussed. The enzyme become denatured. Yes, that's correct one. The enzyme require higher temperature to work optimally? No, it doesn't require higher temperature. It require only the optimum temperature, whatever it is. Lab 4, enzyme activity. Amylase is an enzyme found in the human saliva that can catalyze the production of the maltose sugar from starch. So it means that if you have starch, it can be converted into maltose by the help of amylase. So amylase is an enzyme. So who is the enzyme? Amylase. And who is the substrate? Starch. And who is the product? Maltose. And remember that this product, maltose, can be a reactant for another enzyme. Okay, for example, this maltose is a reactant for an enzyme called maltase. Okay, so maltase is an enzyme which can break down further the maltose. An experiment was conducted to measure the amount of the maltose produced using ml starch of solution at 37 degrees Celsius. Use the table below which shows the data of this experiment to answer question 12 and 14. And this is the table they have given you: time in minutes and maltose concentration in micrometer molar, and these are the values. Identify the dependent variable and independent variable and one control variable for this question, for this experiment. Okay, now what do these mean? Dependent variable is the one that is being measured. Okay, that is being measured. It can vary from reaction to reaction. Okay, so what is the dependent variable in this one? Measured. So look into the statement above, what they are measuring. So they are measuring what? They are measuring the amount of maltose. So what is the dependent variable? What is the dependent variable? Amount of maltose. Please make sure if any unit is given next to it, must mention. It's really very important to score the full marks. Independent variable is anything that you change. Okay, that is being changed to test the effect. And what are they changing over here? They are changing the effect of time. So they are changing the time. So here independent variable is time. So as you change the time, you see that the maltose concentration produced is being changed. Okay, so this is the one. Now let's come to the control. Who is the control? The control is any variable that is kept or that should be kept constant. It should not change. Anything that you know it will affect your reaction, keep it constant. For example, try to recall the major factors we studied just few moments ago. Who can affect temperature? Can affect this process, right? pH can affect this process. Inhibitors can affect this process, but control the inhibitors. Let's remove them. So we can just make sure temperature is regulated, pH is regulated, amount of enzyme. Okay, amount of enzyme. So make sure that you know amount of the enzyme. Also the substrate. Okay, the concentration of substrate, because you cannot say the product is being produced. Okay, yeah, this is also, you should make sure that it's controlled. Plot a graph from the data provided in the table. Okay, you need to plot a graph. The graph is down, so you should know on the x-axis it's always independent variable, on the y-axis it's always dependent variable. Plot it and then make a curve out of it, and you can find whatever you know. Predict and explain the possible changes in the activity of the enzyme for this experiment. If this experiment was repeated at 50 degree, this experiment is being conducted at 37 degree. Please make a note, it's being conducted 37 degree. And we already did one question when they say that what will happen if it is done at 90 degree? The enzyme become denatured, right? So that's the same answer you have to say over here. But what you will say, you can take the answer from here. Look, then add the rate of the reaction did not change by adding this enzyme. Okay, so that's what you can write over here also. The rate of reaction will not change, or it will decrease actually, because you have already there. So rate of reaction will decrease. And why? Because the higher temperature, this higher temperature is, it is more than the optimal temperature, that was 37 degree. So the enzyme, what happened to enzyme? So enzyme is denatured, and it's not binding with the substrate, that the starch. So it's not binding with the starch. Enzyme is denatured, so it do not bind with starch and no product that's maltose, no product that's maltose is produced. But let me guide you one more thing how to answer. Look, the question has two parts, predict and explain. So what we did now, it was explanation. And what is the prediction? What is the prediction on the activity of the enzyme? Okay, so how you predict it? The rate of reaction will decrease or become zero. This is prediction, and here it is explanation why it will decrease or become zero, because the enzyme has been denatured. And that's all for term 1. Inshallah, see you in the next video in term 2 content. Thank you.